So we discovered a novel class of metal drugs that have the unique ability to induce programmed cell death in cancer cells. This form of programmed cell death is also known as apoptosis, a form of cell death that is highly desirable to induce in cancer cells. What makes these class of compounds particularly interesting is their ability to not induce the same extent of cell death in normal cells. We then went ahead and patented these compounds and tested several of them against several different cell lines. In the majority of the cases, these cells induce intrinsic apoptotic cell death, which is particularly exciting given that cancer cells have uniquely adapted bioenergetics. In batch testing, we revealed that these compounds have the ability to affect brain, skin, breast, esophageal, liver, adenocarcinoma, blood cells. Very exciting finds indeed. What we also discovered in toxicology studies is that up to 3 grams per kilogram could be tolerated by rats without being adversely affected. So collectively and taken together, we believe that we've actually stumbled on a unique class of new chemotherapeutic drugs that could potentially aid cancer patients.
So we were working on the crystallography of these silver phosphine compounds, an enormous group of compounds. Crystallography basically means we're interested in the actual shape of the molecules, how they form in a solid state. Now, this was an ongoing process in my research group, and we were interested if there were other aspects to this research. So the nice thing about this group of compounds is if you make them, if you synthesize them in the lab, depending on the actual recipe, you get a different compound. So you get this group of compounds that can be tweaked, that can be changed every time you change the recipe of how you make these compounds. And if you look at the commercial starting materials and calculate the amount of compounds that can be made, we're talking about a huge group of compounds. I'm talking about 3,000 compounds at the minimum. And the best part about it is they're very cheap. It's a very low cost synthesis, it's easy, it can be done in every lab if you know the recipe properly. And the synthesis is pretty reliable, so every time you repeat the synthesis according to the recipe, you get the same compound. Now we took some of these compounds and started looking for additional opportunities, or additional properties. And we were lucky that the first 5 or so compounds that we chose showed anti-cancer behavior. These compounds were killing the cancer cells at a very low concentration, whereas the healthy cells remained alive. What do I mean with a low concentration? We're talking about a concentration that's 10 times lower than the effective concentration of a commercial daily used anti-cancer agent. What do I mean with non-toxic? We're talking about a number of grams per kilogram of body weight in mouse studies, which is equivalent to about 3 grams per kilogram of body weight, which would be the toxicity of vitamin C approximately.
This research is part of a 10-year project by Prof Reinout Meijboom from chemistry on the left and Prof Marianne Cronjé from biochemistry in the middle at the University of Johannesburg. Dr. Zelinda Engelbrecht from biochemistry on the right is the lead researcher for these results in the Biometals journal. To see the research article, follow the DOI link at the bottom of the screen.